Hello everyone, welcome back to Rahman Chemistry Coaching Academy. I am Dr. Taj Rahman and this is our fourth lecture on stereochemistry. In our today lecture, we will discuss and explain the fourth type of structural or constitutional isomerism known as tautomerism. The word tautomerism has been derived from Greek word tato plus meros, which means same parts. Tautomerism can be defined as the isomers that can exist in two readily interconvertible form and who differ remarkably from one another in the relative position of at least one atomic nucleus. Generally, hydrogen atom are known as tautomers and this phenomena is known as tautomerism. The tautomeric equilibration is catalyzed by an acid or base. Now, to explain and discuss the phenomena of tautomerism in detail, let's we first take an example from the hydration of alkynes. This one. This reaction shows that the hydration of alkynes yield an alcohol and this type of alcohol is called enol. The enol possesses an important property of rapid isomerization to an aldehyde or ketone under the condition of their formation. The aldehyde or ketone is called the keto form and the keto-enol equilibration is called enolization or keto-enol isomerization or simply keto-enol tautomerization. The keto-enol tautomerization or keto-enol isomerizations involves the sequence of proton transfers as shown in this mechanism. If we look at to this mechanism, we can easily notice that the first step is the protonation of the double bond of this enol and which is analogous to the protonation of the double bond of an alkene. This protonation takes place more readily because the carbocation form in this step is stabilized by the resonance delocalization of the lone pair of electron present on this oxygen atom. If we look at the two resonance forms A and B, we can easily say that A has 6 electrons around its positively charged carbon atom, while B has 8 electrons each one around carbon and oxygen atom. Thus, B is more stable than A. The keto-enol isomerization or keto-enol tautomerizations involve two separate proton transfer steps rather than a one-step process in which a proton jumps from a carbon to oxygen atom. Furthermore, it may be noted that tautomerization is relatively slow in neutral solution but can be catalyzed either by an acid or base. This one is a base-catalyzed enolization of an aldehyde or ketone in aqueous solution. This is an oral reaction in which aldehyde or ketone is converted into this enol in a reversible process in the presence of a base. This reaction is completed in two steps. These two steps are a reversible process. The first one is slow step and the second one is fast step. In the first step, this base or hydroxyl ion abstract a proton from this alpha carbon of this carbonyl compound, which may be aldehyde or ketone, and give this conjugated base of carbonyl compound. This conjugate base of carbonyl compound is a negatively charged species, and this negative charge can be delocalized to this position, and this pi bond can be delocalized to this position, and hence it gives an annulate this one. In the second step, the water molecule act as a bronze state acid and transfer this proton to this negatively charged oxygen of enolate ion and give this enol and hydroxyl ion is regenerated. This is an acid catalyzed enolization of aldehyde or ketone in aqueous solution. Again, this is an oral reaction in which this aldehyde or ketone is converted into this enol in a reversible mechanism in the presence of acidic medium. This reactions again is completed in these two steps. Both the steps are reversible. The first step is a fast step and the second step is slow step. In the first step, this hydronium ion transfer a proton to this aldehyde or ketone and give this conjugate acid of carbonyl compound and water molecule is formed. In the second step, this water molecule act as a base and abstract this proton from the alpha carbon of this conjugate acid of carbonyl compound and give this enol and hydronium ion is regenerated. So in base catalyzed enolization and acid catalyzed enolization of an aldehyde or ketone in aqueous solutions, enol formation takes place. If we look at these two mechanisms, that is base catalyzed and acid catalyzed enolization of an aldehyde or ketone in aqueous solution, we can easily observe that both these mechanisms consist on two proton transfer steps, that is abstraction of proton from carbon and a transfer of proton to oxygen. The main difference between the two mechanisms is that the sequence of the steps is reversed. The abstraction of proton from alpha carbon is the first step in this base catalyzed enolization, while it is second step in this acid catalyzed enolization of an aldehyde or ketone in aqueous solution. In both these mechanisms, proton abstraction from the alpha carbon is the rate determining step. The enol pumps in acid and base catalyzed mechanism can be easily converted into aldehyde or ketone. As shown in this mechanism, this is an enol and it can be easily converted to this ketone or aldehyde as shown in this oral reaction. If R prime is in hydrogen, the product form here will be an aldehyde. If this R prime is an alkyl group, then the product form here will be ketone. This reaction is completed again in these two steps. The first one is reversible while the second one is irreversible. In the first step, a proton is transferred from this hydronium ion to the double bond of this enol and give carbocation and a water molecule is generated. In the second step, this carbocation transfer this proton to this water molecule and yield a ketone and hydronium ion is regenerated. Generally, aldehyde or ketone are more stable than their enols precursors and can be easily isolated when alkynes undergo acid or base catalyzed hydration. Sometimes students confuse the concept of resonance with the tautomerism. So to clearly distinguish between these two concepts, that is resonance and tautomerism, one should note that three facts may be observed in these two concepts. The first one fact is that tautomerism is an equilibrium between the two isomers which differ from each other in the location of a double bond and a hydrogen atom, and hence are different chemical species. Such equilibrium is shown with a pair of half-headed arrows. The second one fact is that resonance structures shown with a double-headed arrow between them are not different chemical species in equilibrium. They do represent different pi binding patterns. The third one fact is that unlike the tautomeric structures in all resonance structures for a given species, the sigma bonding patterns and geometry are identical. So keeping in mind these three facts, one can easily distinguish between the concept of tautomerism and resonance. So this was all about tautomerism. If you find my video lecture interesting and informative, then please like and subscribe my channel.